Good morning, good afternoon, and good evening to the Pasteur Network community, and welcome to this new monthly seminar. And thanks a lot for being here. I'm Hichem, the coordinator of the Pasteur Network monthly seminars. As always, we will kick things off with our speakers, followed by a Q&A session. Feel free to drop your questions in the chat or during the Q&A session. Just a quick reminder, please, to keep your microphone and camera off during the seminar and to help everything run smoothly. This seminar is being recorded and will be shared soon with the community on the Pasteur Network YouTube channel. And you can also have access to all the previous seminars on our YouTube channel on the playlist monthly seminars. Okay, so today's topic is INRS Armand-Frappier Research Center, a new focus on arbovirus threats to fight against infectious diseases. And today we are very pleased to have with us our two speakers. First, David Chatenet. So very quickly, I'm going to introduce David. David is a full professor and director of the Armand-Frappier Santé Biotechnologie Research Center with expertise in pharmacochemistry, medicinal chemistry, peptide engineering, and pharmacology. David leads cutting-edge research in drug development and biotechnology. His work focuses on designing innovative therapeutic agents and advancing biopharmaceutical technologies. And our second speaker is Laurent Chatenet. Laurent is a full professor at INRS Armand-Frappier Research Center. With the aim to better understand flavivirus pathogenesis and to identify new promising antiviral targets, his research team focuses on the identification of flavivirus host interaction engaged in the viral hijacking of cellular machineries important for the infection. He also works on SARS-CoV-2 and uses innovative in vivo infection models to study viral neuropathogenesis. He is also the head of the containment of the INRS Level 3 laboratory. Okay, now I'm happy to leave the floor to David and Laurent, who will present us the INRS Armand-Frappier Research Center and the potential of the new infrastructure to fight against infectious diseases. And we will come back for the Q&A session. Thank you very much. David, the floor is yours for the first presentation.
Thank you very much, Hichem. Hi, everybody. So before giving the floor to Laurent, I would like to show you a little bit what is INRS, because INRS is a bigger institution than just our center. So in the coming slide, I will just explain to you what it is and how the other components of INRS could also be involved in the Pasteur Network. So what is INRS? INRS is the abbreviation of the National Institute of Scientific Research. It is focused on fundamental and graduate studies and the training of researchers. So we don't have undergrads, we just have master, PhD, and postdoc. So our mission is dedicated to specific goal and particularly directed to activity towards the economic, social, and cultural development of Quebec, Canada, but also worldwide, while ensuring the transfer of knowledge and technology across all sectors. So within INRS, there is the Armand-Frappier Santé Biotechnologie Center that you all know that is part of the Pasteur Network. But we also have three other components, three other centers that are dedicated to specific research area. So we have one in Quebec that is dedicated to earth, water, and environment. One which is split between Montreal and Varennes, which is dedicated to energy, material, and telecommunication. And finally, we have a fourth one that is directly in Montreal and dedicated to everything related to urbanization, culture, and society. Aside those four major research centers, we also have created in the past 2 to 3 years joint research units that are here to consolidate inter-university research and innovation clusters across Quebec. And they are particularly dedicated to specific research area. And the one that is linked to our research center is the one at UQAC, so the second one, which is dedicated to sustainable health. So INRS UQAC Joint Research Unit in Sustainable Health. The other, as you can see, are really dedicated to other area completely outside health research, but quite interesting, with such like cybersecurity, support in regional development, indigenous studies, and material and technology for energy transition. So I would just give you an overview of the different center and how they can be involved in some initiative of the Pasteur Network. So the research center dedicated to water, earth, and environment have 4 different thematics. One in hydroclimatology, where, for example, laboratory dedicated to hydraulic studies or the assessments and anticipation of natural hazards associated with climate change, which is of concern of everyone, and other anthropogenic impacts. Another theme is ecology, ecotoxicology, and environmental genomics, with the monitoring and evaluation of the effect of climate change and contaminant on living organisms, with field sampling, laboratory work, and the use of new molecular tools. The third one is dedicated to environmental technologies, with, for example, water treatment, decontamination of polluted matrices, and the rehabilitation, of degraded sites. They're also kind of interested in the valorization of residue and the development of bioproducts based on CO2 and the sequestration and utilization of this gas. The last one is environmental geosciences, with water resource management, rehabilitation of contaminated aquifers, agroenvironments, and coastal erosion. There is also 2 last topics on geoscientific data processing based on artificial intelligence and the use of multi-isotopic approaches in water geochemistry. So with all the problematic related to water use and the water pollution, that could be a good way, to use this center in your initiative. The other research center, which is maybe a little bit far away from what we are doing at the Pasteur Network, is urbanization, culture, and society. They are mostly involved in everything related to evacuation, trauma, anxiety, PTSD in the social way, how conflict and migration is also impacting the health, and everything related to the social and structural determinants of health, with specific area of research like the family, including issue of public guardianship, fertility, desire for children, and co-anxiety. Everything related to adaptation to climate change in urban area, including health effects. They have also created an observatory on youth, including their physical and mental health. They have a specific area of expertise in anti-racism and decolonization. And they also have developed an expertise in dynamics of urban environments. So it's a little bit far away from what we are doing inside the Pasteur Network, but I just wanted to show you that we have also some kind of tool more in the health population. The third research center is based on energy, material, telecommunication, but I will mostly focus on everything related to energy and material that is quite easily linked to everything related to health science. Yeah, no question. Okay. So here I'm going to show you 2 slides with kind of different expertise and how they use their specific area of knowledge to develop, for example, here, DNA nanostructure to prevent the formation and convert toxic amyloid protospecies into cytocompatible and biodegradable spherical complexes. So for those who are not aware of what is amyloid, it is involved in Alzheimer's and Parkinson's disease. So using DNA nanostructure, they are able finally to prevent the aggregation of amyloid and finally reverse the pathology, the complexical pathology. Another example, which is maybe more related to what we are doing in the Pasteur Network, is the rapid and systematic screening of bispecific antibody surrogate geometries for T-cell engagement, again, using DNA technology. So this might be quite of interest for people developing new vaccine based on antibody. Here is the slide is a little bit charged, but I'm going to show you what is really interesting for us. So another way to use kind of what we call rare earth material is to develop light-controlled drug delivery system. Also photodynamic therapy, for those who are not aware what it is the use of light to trigger a response with near infrared and to kill tumor cells or to kill maybe bacteria. That could be another way to use that technology. So they use rare earth material to develop such kind of photodynamic therapy-based compound. So there is also the photothermal therapy. Again, it is the use of rare earth material and the difference in temperature within a cell or within a tumor to specifically kill such tumor or such cell. There is also near infrared, bioimaging that could be used, to look in vitro and in vivo for application in cancer, but also inflammation or so on. And the last one is the nanothermometry. It is a way to measure directly within the tissue or directly within an animal differences in temperature in particular specific area of the body, which could be related to inflammation or an increase in metabolic activity. So this is finally what is done in these other three centers. So I'm going to go back to our center, the Health and Biotechnology Center, with four research theme. So one is host pathogen interaction. The second one is action of xenobiotics and genetic variation on health. The third one is the development of therapeutic, preventive, and diagnostic agent. And the last one is epidemiology and biostatistics. But all of those four major research teams are engulfed in the One Health concept, since we are working in human health, animal health, but also environmental health. So we are targeting those three major components of health research. In terms of research training, I know that you might be interested in that. So this is to show you that at our center, we have a free master program, one in virology and immunology, one in microbiology and biotechnology, one in experimental health science. This particular one has different concentration that you can see in the right panel, and concentration on host pathogen interaction, biomedical diagnosis, epidemiology, pharmaceutical chemistry, bioinformatics, and pharmacology and environmental toxicology. So that could be also a huge opportunity for students from abroad to come to our research center to get those diplomas. We have also two doctoral programs, one in virology and immunology, and the second one in biology. What will be new in 2025 is we're going to have an entrepreneurship course that will be given within our center, but also we are planning to launch a management of preclinical studies course in collaboration with our animal facility and maybe a company, Child Reverse, that might be able to offer some internship to have hands-on expertise or experience in the management of preclinical studies. So the research team in our center, I'm going to go kind of quick. So the one which is related to the action of xenobiotic and genetic variation on health, you can see on the right, this is pretty much the area of expertise that we have. So we are working on Alzheimer, ALS, Parkinson. In the cancer research area, we are targeting breast, liver, prostate, and ovaries cancer, but also rhabdomyosarcoma, which is a cancer related to muscle. We also have an expertise in reproductive toxicology, more specifically male infertility and everything related to pregnancy. The epidemiology and biostatistics research team is oriented to different perspectives, including the trajectory analysis, personalized medicine, and the application of causal inference method in the context of case-controlled studies, the prevention of prostate cancer, or the link that are related to an increase of this specific cancer, the study of factors influencing the immune response and their role in the etiology of various cancer, but also infectious disease, study of the lifestyle habits, effect of different environmental contaminants on population health, but also the development of machine learning methods for better modeling of genomics and brain imaging data. So this kind of expertise that we have here, so do not hesitate to contact us if you see any potential collaboration. Another research team is based on the biotechnology and food science. So we are working on what we call the microbiome environment. So we are looking at the bacterial population and how this population is involved in the agriculture and the growth of the plants. We are also working on what we call soil remediation. So how we could implement new bacteria or complement bacteria that are non-existent or should be present to again help the growth of plant and the quality of the plant. We are also interested in what we call circular bioeconomy. So it is the use of, let's say, waste product and how we can valorize this waste product to have the minimal waste at the end. So instead of burying this waste material, is how we can use them again in the process chain and have minimal waste at the end. The last one is food science. So how we can make food better with a better quality in terms of specificity. But also it is how we can use bacteria as biopesticides to help finally manage different kind of disease related to, in the example in the right, tomato, for example, but it could be corn or other type of crops. The last theme, which is related to the presentation of Laurent just afterwards, is everything related to host pathogen interaction. So you have a non-exhaustive list of what we are working on. So you have the viruses like the arboviruses, you have parasites, leishmania, and the bacteria with Pseudomonas aeruginosa, Neisseria, Mycobacterium tuberculosis, and Escherichia coli. The last theme is the development of therapeutic, preventive, and diagnostic agents. We have a good amount of researchers that are working in different kind of area. And you can see here, for example, we are one of the group here at the, we have one group, sorry, here at the center that develop new tools, non-murine animal models, to study different kind of pathology. It could be a virus infection, but it could also be cardiology, or any neurodevelopmental diseases. So I think in the context of what we are seeing right now in France, when you can have a tax now on the use of murine animals, that could be an interesting model to continue the development of different therapeutics. This non-murine model, which is a zebrafish, could also be used as a toxicology model. It is approved as a toxicology model. We are also developing different kind of organoids, more specifically breast cancer 3D organoids, to again look at different kind of imaging modalities, but also as a model to test new drugs. In terms of chemical expertise, medicinal chemistry expertise, we have here structural biologists, peptide chemists, organic and inorganic chemists, but also carbohydrate chemists. So we are kind of covering all the different areas of the chemistry, which help us finally to bring our expertise in different possibility for the biological group here at our center, but also abroad. We have also an expertise which is kind of interesting in the fragment-based drug discovery, which is the use of NMR, so nuclear magnetic resonance, to identify binder of specific proteins and at the end design drugs that are completely new and could lead to new IP. In terms of area of expertise and area where we are using these experiences, we are specifically developing new therapeutic and diagnostic agents for cancer, cardiovascular diseases, Alzheimer's, Parkinson, myelioidosis, but also antimicrobials and antibacterials. The last one is the use of non-coding RNA, so what we call riboswitches and ribozyme as biosensors. So as you may know, ribozyme and riboswitches are RNA-based therapeutic tools that are most often employed to knock down gene expression and to inhibit bacterial infection. So we have a group here that is working specifically on the development of this kind of riboswitches and ribozyme. I'm going to finish to make the transition to Laurent with the different infrastructure, because research cannot be done with state-of-the-art infrastructure. So we have at our center an electronic microscopy platform, confocal microscopy platform, BCL2, BCL3 facilities, and a biophysics platform that encompass, circular dichroism, nuclear magnetic resonance, internal calorimetry, and mass spec for proteomics and so on. And also we have what we call the National Experimental Biology Laboratory, which is a certified animal facility for everything related to animal care here in Canada. But we have also an accreditation that let us finally work with different university or company outside Canada. So our animal facility is kind of big because we are offering housing services for rodents, zebrafish, rabbits, non-human primates, dogs, chicken, chicks, but also large animals like sheep. So that's also another expertise that we could bring to the Pasteur Network. So I'm done with the presentation. I will be happy to answer a question at the end of the talk of Laurent. But right now, thank you for your attention, and I'm giving the floor to Laurent for the next part of the presentation.
Thank you, David. Laurent, okay, perfect.
Yeah, so hello, everyone. As David mentioned, I will show you today, explain you today the latest efforts of our center to develop a pipeline to study pathogens, mostly in containment level 2 and 3 conditions. So I just wanted to make a little bit of background, and you heard the name of Armand-Frappier, and why I want to talk about him today very briefly is that he was a visionary and a pioneer in biomedical research in Canada. And his focus was mostly on infectious disease. And why I want to talk to him about him today is that he was often referred to as the Quebec Pasteur. And the reason for that is actually went to Institut Pasteur Paris and then brought back the BCG, the tuberculosis vaccines, back to Canada, and then could provide it to the population here, including the indigenous people communities in the far north of Quebec. And of course, he's the founder of our research center, and it was really based on the Institut Pasteur model, where the center would biomanufacture some vaccines for the government, for the public health, and that would in return support the basic research in our center. But now you can see that this historical bound with Pasteur is still maintained since we are part of the Pasteur Network since 2005. So I just wanted to make this kind of a historical link between the Pasteur Institutes and our center. But now we're still working on infectious diseases. It's one of the main themes of our center. We work on many viruses, not only arboviruses. This includes HIV, herpes virus, flu. And it's really, it comprises two of the three research themes of our program, which are infection immunity and development and therapeutic and prophylactic agents. And recently, maybe 2 years ago, we launched a research network, which is still being built, which is called the infectious INRS. But why I want to talk to you today is about our containment level 3 facility. In the center, we have a CF3 lab where we do what I call in cellular studies. I mean by that cell culture, when we do infections of cells and do analysis. Right now, we are certified by the Public Health Agency of Canada to work with SARS-CoV-2. We have several variants of concerns growing in the lab. This is also one of my main interests to work on West Nile virus. It's, I don't know if you know about it, but it's related to dengue and Zika virus. It's transmitted by mosquitoes. And we're also certified to work on tuberculosis. And of course, our aim is to expand this portfolio and get a certification to work on other viruses, including, of course, arboviruses, my main focus. We also have an infrastructure for containment level 3 animal facility at the National Laboratory of Experimental Biology that David just mentioned. So this is meant to work work with animals, mostly rodents. It's still not certified, because now we have some issue with major renovation in this laboratory. So we had to move temporarily the animals to the BSL3 infrastructure. So now it's in containment level 2. But as you can see, it's pretty large. It's more than 600 square meters. There are 4 sync rooms. So we hope that in the very soon, maybe in 2 years, this will get certified by the Public Health Agency of Canada, and then we'll be able to work with animals in a CL3 condition. And this is really important for us because this is part of a larger effort from our center to build a pipeline to work on arboviruses at all levels. So, of course, amid the COVID-19 crisis, the government of Canada and the government of Quebec invested a lot of money for pandemic preparedness. And then at that time, we worked a very ambitious project that was focused more on arboviruses and not like respiratory viruses, which was the main focus of many universities. So we wrote this project, and we were very lucky that in February 2024, the Ministry of Economy, Innovation and Energy of Quebec funded us with more than $4 million to acquire equipment for our BSL-3 infrastructures. So this pretty ambitious project relies on three main objectives. The first objective is to really expand our portfolio of risk group 3 pathogens with a focus on arboviruses, but not restricted to that. And one of the issues we had is that for now, we cannot work with non-endogenous pathogens. There is special regulations when want to work with pathogens and or arboviruses that do not circulate in Canada. And that's because it's not because of the public health of human, it's because the Canadian Food Inspection Agency is really very protective about importing any pathogens that do not circulate in Canada because they want to protect the livestock. There are like many economical consideration related to that. So for instance, for that, we need to take a shower and everything. But we have the infrastructure, except that now we do not have we have order. When one wants to work with non-endogenous pathogens, the decontamination of liquid has to be better taken care of. That's why with this funding, we bought a liquid waste decontamination system that will allow us to expand this our portfolio of viruses to work with in our infrastructure. So we have ordered that. It's supposed to be installed in March 2025. So with that, we could work with, for instance, Japanese encephalitis virus or Uzutu virus. Uzutu is BSL-2 in Europe because it's circulating, but since it's not circulating in Canada, we have to have extra care about that. That's just an example. And of course, one of the main focus of this laboratory is to work in culture cells that we will infect with risk group 3 pathogens. And we bought this equipment, which is called a Citation C10. It's a big plate reader. It allows us to make classical experiments like fluorescence measurement, luminescence, optical density. But the real power of this machine is that within it, there is a confocal microscope, actually a spinning disk microscope. That means that there is a possibility to perform high-resolution microscopy on living cells that are infected with risk group 3 pathogens with a very high resolution, and it's very fast. We can do it in very small formats, so acquire many wells of a plate. And also we have a module that allows us to do medium throughput screening. So simultaneously in one experiment, we can screen about 5,000 conditions. 500 conditions, sorry. No, 5,000 conditions, sorry. And this means that, for instance, there is a possibility to screen drug libraries. It can be image-based, or it can be luminescence or fluorescence-based. This is doable, and we can also perform genetic screening using RNA interferon under the CRISPR-Cas9 technology. In parallel, we have also acquired ultracentrifuge. This is quite classical, but we didn't have it, and for us that was an issue. And that means that now in infectious conditions, we can concentrate viruses, for instance, or we can, if we are more interested in the life cycle of viruses, we can purify organelles from infected cells. For instance, mitochondria, and then we can isolate those organelles from the cells, then study them. Or the nuclei for the DNA, for instance. We have also acquired a new confocal microscope from Zeiss. This is the LSM 980. The very interesting thing about this device is that it has an Eriscan module, and that will allow us to increase the resolution of our microscopy studies. We will be able to do super resolution studies here. You see an example. This is a mitochondria from Arabidopsis, okay? you can see that one do not get good resolution in classical confocal microscopy because it's too small. But then when with this Eriskin module and the joint deconvolution modules, then you can see what kind of images can be acquired. That means we can really dissect what's happening in infected cells for whatever we study resolution that we have so far. So we can really go deeper into the world of the small things. This will allow us also to do live-cell imaging in infected cells. I have to say that this microscope is in containment level 2 conditions, so we will be able only to use risk group 2 pathogens. And this is partly due, it has a very fast scanning capability and it's highly sensitive. We have also acquired a flow cytometer and analyzer from Santé, and the power of this is that it allows to do full spectrum cytometry. It's not based on you look at one marker at one given wavelength. You can really address the full spectrum of wavelength, and this allows you to add multiple markers, many more markers. The nice thing also is it has an automated loading of samples. Basically, you can put your plate of 384 wells, put it in a machine, and then go. It will do it. It will load the samples automatically. So, sorry, the spectrum, the throughput of this machine is much higher than classical analyzers. Those devices have been received. They will be installed in early January 2025. I think the Microsoft might be installed a bit earlier. Okay, and the third objective of this project was to implement a multi-scale pipeline. So we really wanted to analyze infection with a given virus that would emerge, like a new arboviruses, and then put it in an animal, and from that animal go to the analysis of one single cell at the transcriptomic level. So first, we wanted to do in vivo imaging, so to look at the animal and see where the pathogen could be, how it spread over time. For that, we have acquired an IV spectrum CT from Revity. What this allows you is to, for instance, if you have a virus that expresses luciferase, then you can detect in situ the luminescence, and the power of this is that it has this X-rays, and you can detect the skeleton, and then you can do 3D imaging. So basically here you could see, for instance, a dengue virus that expresses luciferase in the liver. And then you can really quantify this. And what is really cool is that the mice are still living. They are just asleep. And that means that you can do that like every 2 days and see how a pathogen spreads from the blood to other organs, and then how it moves to the brain, and then test antiviral molecules, for instance, and see how this impacts the spread and the amount of pathogens within the animal over time. You can do that also on isolated organs if they are perfused, so that's really cool. And this is just what I just say. Actually, Frédéric Verrier, which was part of this proposal, do that on a regular basis at the 2D level with for meningitis. Some people do that also to visualize cancer cells within the animal. So this device is located at in our animal facility in containment level 2 conditions, and Frédéric Verrier is responsible for it. It's part of the platform for the characterization of biological and synthetic nanovehicles. Basically, it's our electron microscopy facility. And then once you have your infected animal, of course, you want to study the pathogenesis and then to analyze relevant organs that could be infected or not. So I gave 4 examples of extraction of blood, liver, spleens, and brain from infected animals. And then, of course, what is often wanted is to study a particular and specific population of cells to see how they react to infection. Are they infected? Do they produce cytokines? Are some pathway stimulated? Stuff like this. So the idea is to really fish those specific cells. There are also many subtypes of lymphocytes, for example, or many subtypes of neurons, and to see specifically in those rare populations how infection modulate their function. For that, one needs to enrich for cell populations, sometimes very rare, and then analyze them, and in that case, at the transcriptomic level and single cell transcriptomic levels. For instance, what kind of subpopulation of neurons are impacted during Zika virus infection, for instance. Sorry. Okay, for that, we have acquired a flow cytometer, which is a cell sorter. This allows to sort cells and to get them back and to analyze them. They're still alive, or you can put them in culture if you want, infect them, do whatever treatment you want. Again, it's a full spectrum flow cytometry. So in theory, because of the spectral deconvolution, it is possible to analyze up to 40 parameters simultaneously. So one can imagine that we can really fish very rare cell population. That's pretty unique in my opinion. And keep in mind that again, afterwards the cell are still alive. So it's not just an analyzer, it's really a cell sorter. It has 5 lasers. It's in a BSL2 biosafety cabinet in level 2 for now, since our animal facilities is not certified for CR3, so we keep it in level 2. I put what we have as nozzles, but the point of that is that we can also sort very small particles. It means that within cells, we can isolate, for instance, mitochondria, and if your virus of interest is above 70 nanometers in diameter, then in theory, one could sort viruses as well, virus populations from the cell supernatant or blood, or even from the cell. So once you have your rare cell population of interest, the idea is to partition them and to do single cell RNA sequencing. For that, generally, first, one needs to do cell partitioning before preparing the CD in the libraries. And for that, for the partitioning, we have acquired this Rhapsody. So basically what it does, it separates the cells individually that you have sorted before. It takes one cell and then put some primers, barcoded primers for the single cell sequencing. This is also in a containment level 2 biosafety cabinet, so that we can do that on infected material with risk group 2 pathogens. Again, we are waiting for the certification of the animal facility for the level 3, so that we can do that in level 3. This is installed in the laboratory of Alain Lamar in our center, and the cell sorter is also, as well as the microscope, are located at our core facility for confocal microscopy and flow cytometry of our center. So, okay, I told you about this pretty nice pipeline, it's a lot of machines, but how is it relevant for the Pasteur Network? How can we connect this infrastructure to the expertise of the Pasteur Network mission? So, as I mentioned before, this infrastructure project was primarily designed or focused on arboviruses. I thought there was an interest to study those neglected viruses, at least in Canada. So for now, we use them to study dengue virus, Zika virus, West Nile virus. We plan to work on chikungunya, also Eastern equine encephalitis virus, which is relevant in North America. And of course, the idea is for preparedness, right? Is to respond to the emergence of what I called arbovirus X that we don't know yet. I've been a bit provocative, and that could cause disease X, the famous disease X of the WHO, but it's a bit provocative. But of course, this is not restricted to arboviruses. This pipeline is open for other relevant pathogens, whether it is bacteria, respiratory viruses, or parasites. And then when look how it could be connected to the mission of the Pasteur Network, I look back to the Pasteur Network website, and those are the 4 pillars of the mission. And of course, the most obvious connection are to those 2 pillars: the epidemic intelligent preparedness, focus on climate-sensitive diseases. So this is the case of those diseases. You all know that much better than me. Also, it's clearly connected to the research development and innovation pillar. But the idea is, this is quite obvious, but how can we make this infrastructure also relevant for the three other pillars? All right, how can we bring it to multidisciplinary research, and how can it be accessible to other members of the Pasteur Network, which maybe do not have access to it now for now? And I also noticed, I'm well aware that the Pasteur Network is building those new scientific working groups, and I think it could be relevant for the vector-borne disease impacted by climate change group and also the epidemic preparedness. So I think this is all relevant for the Pasteur Network mission. But then you might, sorry, my cat is aggressing me. Then you might wonder, well, this guy is in Canada. He talked to us about the dengue, Zika, but why? Why is he working on that? And why is it relevant for Canada beyond just a general contribution to the global effort to research on basic principle of virology? I guess some of you, when they think about Canada, they see this. They see a lot of snow. You have to rediscover your car again upon winter. Those crazy temperatures, which is not the worst actually that we see on a regular basis. Let's say in January or February. I'm pretty scared about this, but actually I like it. We are located here, so you see Quebec is quite large. We are pretty north in America, so of course dengue might not be seen as relevant here. But the reality is that this is a continental country. Winter has something, but summer also, and we have a lot of water, and that means that we have a lot of mosquitoes, a lot, and of course, since there are many mosquitoes, this comes with the disease they carry. I mean the pathogens they carry, and in that case, arboviruses. West Nile virus is actually circulating in an endemic manner since 1999. West Nile virus, for those who do not know, this is a flavivirus. It's closely related to dengue virus and Zika virus and yellow fever as well. We know that St. Louis encephalitis virus and the Eastern equine encephalitis are circulating in Quebec. There are not so many cases, but they are circulated. And when one gets infected by those viruses, the consequences for the health can be pretty severe. And one of the issues that there is no surveillance by the Public Health Agency of Quebec since 2022. There is no money for that, no interest. This is mostly due to that all the resources have been redirected to COVID-19 during the COVID-19 crisis. And of course, now the health agency has to convince again the government to inject money in those diseases. But the point I wanted to make here is that climate change is also relevant for Canada, actually. I can tell you that winters are not the same for a couple of years now. The temperatures are making the yo-yo during the winter. We get a lot of rain, which we didn't have like 20 years ago. So it's also impacting Canada. The winters are warmer. Actually, the Aedes mosquito have been detected a couple of times in the south of Ontario, which is here. It's a little bit warmer. But that means that, as in Europe, those mosquitoes are actually colonizing northern territories. I mean, more temperate countries, and this relates also what is seen in the US. Those mosquitoes are coming up, like in Europe. And also in Latin America. I mean, seeing what happened with dengue last year in Argentina, for instance, or Uruguay. So actually, arboviruses are quite, I think in the future might be relevant for Canada. And I mean, I always like giving these symbols. I agree, it's totally science fiction, but one might think that with the melting of the permafrost, that might release some ancestral pathogens. I mean, we know that there are ancestral viruses, giant viruses in the permafrost from Siberia. So it could be the case here. And some communities, indigenous communities live here. So there might be a risk to be, might be at risk of those new pathogens, resuscitated pathogens. So the bottom line is that I think and that's a personal opinion, but arboviral threats matter everywhere, not only in very warm countries or very humid countries. It's also, I think, everybody will face those threats again. And I wanted to go back to the initial question I asked a couple of slides ago, is how can we connect this infrastructure and the associated expertise to the Pasteur Network mission? So, of course, this is a classical view. We can collaborate. I mean, I'm really willing to collaborate with the other Pasteur Network research centers. And we have a technical expertise for studying fundamental concepts of arbovirus biology. There could be an access to those technologies for some region that might be more focused on public health and may not have the infrastructure to do molecular virology. And of course, the obvious things that grants can be written, we can get money for collaboration. ICPD are the most obvious ones, but there are other possibilities. But one thing that is very important to me, which is one of my goals, I do molecular virology, mostly fundamental virology, and I'm always wondering how does this connect to the reality of the field? And maybe in some of your region where you work, when you have field challenges, you might have questions that you cannot assess. So I think this is where we could do multidisciplinary research, can be anchored also to surveillance, to also social issues, I think. You get sequence, we can test them, but it has to be in a mutual interest and benefit. The idea is not that we get samples from some people. The idea is really to work together, that we can also answer questions that you're interested in. And on our side, we are making a lot of effort to anchor this to the field in Quebec as well, to obtain local clinical samples, for instance, from people who recover from West Nile virus to study their neutralizing antibodies, for instance. We are also getting samples from birds who died from West Nile virus. We are trying also to connect with the Public Health Institute of Quebec about mosquito surveillance. They used to do that, but as I said, it's quite challenging because they don't have funding right now for that. But we are in talks to work together, but that's not an easy task, but we are working on it. And one very important thing, I think that could be one of the main contributions of this infrastructure to the Pasteur Network, is that actually, I think it would be quite feasible to train some researcher or personnel of the Pasteur Network, for instance, to learn how to work in the containment level 3 laboratory, so to do some training, but also to test their question with our equipment, or to learn some molecular arbovirology techniques that we have if we have some reverse genetic system. So I think we could pretty much easily welcome people from around the world for a couple of months, let's say 3 months. It's much easier regarding visas, but I mean, everything is open, or I mean, your feedback would be much appreciated, but I just wanted to let you know that we are pretty much willing to host people who wants to learn those techniques if that's relevant for them back in their region. And of course, we are really willing to participate to different working groups implemented by the Pasteur Network. So as I said, there is those scientific working groups that are being built, being implemented. I think that could be relevant that we participate for the reason abroad. So I'm looking forward to discussing with you about those arboviruses. And also, Oscar has initiated a discussion in the Pasteur Network members of the America about emerging diseases, for instance. So I think this is very relevant, and as part of the the most north country in Americas, like beyond the world, as some would say, we are pretty much enthusiastic to participating to those discussions. And why not building an anti-arboviral task force? I mean, that would be super cool and super relevant for everybody. But let's create some synergy. I think that's very relevant. And I think also Quebec could be kind of good model for studying the impact of climate change on the infectious diseases. So I know Brazil is in the center of a modeling for the impact of climate change to predict dengue emergence, which serotype will emerge where next year, for instance. So Quebec could be interesting because we do not face those threats right now, but it could be a good model to study the other side of the spectrum and to predict what could happen in the future, especially if the situation evolves here regarding emergence and changes in the climate. And I thank you for your attention, and any question will be most welcome. Thank you.
Thank you very much, Laurent and David, for your presentations. We are a little running out of time, so we will take 2 or 3 questions, maybe a little more if you are ready to stay. So the Q&A session is open. The floor is yours to the Pasteur Network members. We had a lot of info that could be discussed. Oh, someone is talking. Okay. Who has a question? Ilian from the Stefan Angelov Institute. Go ahead. I don't know if you can hear. Maybe you're muted. Oh. Ilian, can you hear us? Well, maybe Claudia Nunes, and we come back to Ilian after that. Claudia. No. Okay. You're muted. I don't know if you're trying to speak. Claudia. Okay. Who else? Maybe it's me. David, Laurent, can you hear us? Can you hear me, actually? Yeah. Okay, so we go to Sonia. Sonia from Tunis. Sonia.
Hello, everyone.
Just to check.
Thank you so much.
And mute your... Yeah, go ahead. Hello.
Yeah, thank you so much for your presentation. Do you hear me?
Yes.
Actually, I have a question about the 4th pillar in which we might find some collaboration, and I had the chance to work with Hichem on science and society aspects. So I wanted to know if, Laurent, in your research activities, do you involve citizens in your research for surveillance, for example? Thank you so much.
So for now, we do not do surveillance. This is because in Quebec, the surveillance is the duty of the Public Health Agency of Quebec. And as I said, the problem is that do not do surveillance again. So they are well aware that we're interested to participating to those to the surveillance system if they do it again. And there are some committees where citizens are involved in the decision taking, or at least consultation. And we would be part of those committees, but this is still, they are working very hard to get more money from the government to study that. But this is not our mandate right now. We are also trying very hard to get clinical samples and to implement, maybe to have a cohort in that case. One of the objectives would be to make, to have patients involved, for instance, in those committees. But for now, I mean, I do purely molecular biology and work on animal models. So for now, there is no citizens involved.
Thank you, Laurent. Back to Ilian. You go ahead. I think you have a problem with your microphone or something. We can't hear you. We can't see you, Ilian. But maybe you can ask your question in the chat. And Claudia, the same. You can just write down your question in the chat, if that's okay for you. Any other question? Renaud, go ahead, please. Can you introduce yourself? also,
Yes,
please?
sure. I'm Renaud Vatrine. I'm from the Pasteur Network office in Paris, and I'm working on training and education activities and as well the scientific working group. So thank you very much, David and Laurent, for both of your presentations. Just wanted to say that it looks like there's quite a lot of interest about those scientific working groups. I just wanted to reiterate the fact that applications are open, so everybody is free to apply. The deadline is the 15th of January, so please do apply. And if you did not get the information, please just don't hesitate to write to me and I will send it again. And as per the training activities, I mean, we've discussed already, and we hope that we will build some great collaboration for the Pasteur Network together. So this is something that we've discussed with Kathleen, and this is ongoing. I was wondering about this entrepreneurship course. How will it be open to the Pasteur Network? Will you have something online to propose? Is there any? And is it entrepreneurship? What does it mean? Is it how to work with the private sector? Is that about technology transfer?
So for now, it is not online, but I'm pretty sure we could manage to do it online and to open it to the Pasteur Network. The way it is built in 3 phases. The first phase is the student will learn about more the theory of entrepreneurship, everything which is regarding governance, legal aspects, intellectual properties. The second phase will be more practical. They will be linked to a researcher here, for example, at the center or at a different university that just want, to transfer his technology to the higher level, but he has no time to do it. So those students will be involved in those projects and they will be involved in bringing the technology to a maturation. So practical, and after that, more theoretical and more practical. And at the end, there will be a certification to demonstrate that they have business management, research, human resources management abilities and everything. So it's the whole package.
Okay, thank you. Excellent. So happy to talk about this initiative later after that it happened and maybe work at for the Pasteur Network. Yeah, thank you.
No problem. In January, if you want, we can do a small talk and I will explain in more detail what's going on.
Okay. Yep. Thank you.
Thanks. Thank you very much. Okay, I have so Ilian wrote the question. Okay. Okay, he asked for training, but I don't know if you're asking for specific training. Ilian, he's just interested to collaborate with you. So Ilian, he's in charge of the animal facility here. I'm saying here because I'm in Bulgaria. At the Stefan Angelov Institute, he's in charge of the animal facility here, and he would like to develop more collaboration. So maybe I can put you in contact together with Ilian. Oscar, I think you raised your hand, right?
Hi, yes. Good morning, good afternoon, good evening. Hey, David, Laurent, it's good to see you. Thank you for the presentation.
Yes,
very interesting. Just a quick question, and maybe of interest for the rest of participants to the seminar. So, in terms of collaboration, what type of collaboration you guys have with other agencies in Canada or in your region or internationally? I was just curious to know what type of collaboration, or if any, do you have, or with other research institutes in Canada?
You want to start, Laurent? I'm going to go.
You can.
going.
So,
okay.
So for that kind of project, we don't have any. We are trying to make one it's related to all the challenges I told you about. So we are trying to put arboviruses in the radar. But regarding other projects that we have many collaborations with, maybe David, go ahead for the others.
Yeah, so we don't have hospital linked to the center. So we are not a university linked to any hospital, but we are kind of developing such kind of collaboration to have a sample from patients. We have also developing collaboration with the public health agency, like Laurent was telling you. But we have also implementing a collaboration with a Q2 solution, which I believe, if I'm right, has already connection with the Pasteur Institute of Dakar in Senegal. So that's something also that we are trying to implement to help them answer questions from a Pasteur Network Institute. So Q2 solution or Nexelis, maybe you heard about that. We are also working with a collaboration with Moderna to have access to their production facility, which is less than 1 kilometer from our center. So we are kind of building the relation with them to maybe build some project, and they could have access to our institute. So that's pretty much it. Internationally, Institut Pasteur, INSERM, INRS, we have some connection there also, but it's mostly local and national.
So what I wanted to add, we don't have a formal affiliation to a hospital or to public health agencies, but regarding specific projects, let's say in epidemiology, our epidemiologists collaborate with the Public Health Agency of Quebec, for instance. So it's really specific to some projects. Some people have access to cohorts of patients that are HIV infected. For instance, there is a huge network for that. Some people work with the provincial agency for the security of workers. They have projects for that to enhance the security while working. I don't know how to say it in la santé sécurité au travail. So there are many collaborations, but it's really project specific, I would say.
Thank you. Thank you very much. I don't know if there is another question. I'm looking for the... We are running out of time, so maybe we could stop here. I had a question actually, but I don't know if you have some... Do you agree if we ask a last question, but it remind... Okay, I just want to have an idea of the type of collaboration that you are having with the Pasteur Network members actually. And do you have, are they mostly ASIP or PTR? Do you have any type of project, and funded by who exactly?
So you're talking about this arbovirus pipeline, or more generally in the center?
No, I mean in all type of collaboration with Pasteur Network members.
I mean there is a we have actually. It's okay, David. We have many
go ahead.
Pasteurians as professors. Former Pasteurians as professors in our center, Frédéric Verrier, Charles Calmette, they got like several PTRs. Frédéric Verrier, I don't know if he's around, is actually has I don't know, an affiliation with the Pasteur Institute in New Caledonia. He goes there pretty often. I mean, depending on the situation there, I know he's supervising a small group there. He works mostly on pathogenic bacteria. So I know that Albert Descoteaux in Océan collaborate with some people in Latin America at Fiocruz. So we have like, again, this is really project specific, but we got like several PTRs and ASIPs, more PTRs than ASIPs. So some labs are pretty well connected with the Pasteur Network community, with Madagascar as well. So yeah, maybe I miss anything, but in my case, I don't. Okay, so this is my will to try to be more connected to the people who face dengue virus, for instance, or to work on West Nile in the Antilles. So this is the kind of thing that I'm really willing to expand, because maybe I face a crisis in my research, but I would like to be a little bit more grounded to the reality of the field and how I also can help the people who are facing that.
Yeah. If I can add, to complement Laurent, so Frédéric Verrier is also involved with LEPSNet. I think it's an initiative from the Pasteur Network in leptospirosis. Albert Descoteaux is involved in the Leishmania, also another Pasteur Network initiative on the Leishmania. And even if I am a medicinal chemist mostly working on cardiovascular disease, I have an ASIP with someone from Uruguay, Nicolas Arrute, and Nolwenn Jouvenet from Institut Pasteur de Paris. So like Laurent was saying, we have different kind of PTR, more project related, because not the whole institute is working on infectious disease. We don't have one goal. As I presented you, we have different kind of theme, but sometimes those themes intersect with the Pasteur Network objectives.
Okay, so I thought it was the last question, but there is a very last question this time. I'm sorry if you can give some time. We have a last question on vaccine development. So from Gita Amadou, thank you, Gita. She's asking, do you have vaccine development programs in your? So I suppose yes, because you're Biotechnology Research Center. But again, can you give us an idea?
So
Go ahead.
we don't have a program on vaccine development. We have some courses, but we have some groups that develop new vaccine technologies. We have, for instance, Alain Amar, who's developing a new vaccine platform. So there is an interest for some people to develop vaccines and maybe to collaborate, of course. I don't do that, but it's really a little bit case by case situation. But we don't have a program of vaccine development. It's more like a research based. So maybe it was not entirely clear, but our research institute is not really dedicated to provide services, public health services to the government. It's really a fundamental research institute, I would say, and also epidemiology. So
in the past,
sorry?
Yeah. No, go ahead. I will just implement after that. Okay, I was just saying that in the past, we were an institute that were producing vaccine, but we were making money, and in Quebec, a university cannot make money. So the government decided to separate the activity of the university and the production side. It was 25 years ago. So like Laurent was saying, it is more now vaccine-based development on the projects. So viruses, but also we are using nanobodies to develop new kind of vaccine for cancer. That's a new opportunity, but we don't have a vaccine development program.
Okay, thank you very much. So I think we're going to end here our seminar. Thank you. Okay, this is my first meeting. How often do you have this meeting? So it's a monthly meeting, Andreas. So and actually, thank you for the question, because I'm going to inform you about the next meeting. So the next one will be on January 16, and it will be with someone outside the network, who is Manu Prakash. He will tell us about frugal science and how to use different type of tools on the field. I hope you will be many of members of the Pasteur Network able to be here. Okay, so I wish you a great end of the year. Merry Christmas, Happy New Year to everyone. And thank you again to David and Laurent for your intervention. I hope it will open many new collaboration between the INRS Armand-Frappier and the Pasteur Network members. Thank you very much. Bye-bye.
Thank you.
Thank you.
Bye-bye.
Bye-bye.